Okay, it's really, really nice to be here. It's really nice to be first, actually, and it's really nice to be part of the first edition of a festival. And just speaking with Flora and Vera last night, I was really struck by the ambition that you have with this, and I think it's really cool to be part of a set of people who've got a bit of a vision for change. So today I'd like to talk to you a little bit about something that we've been thinking a lot around in UCL, which is this idea of the Biocene. So at the moment we have this, if you like, a doom loop of how we think and talk about climate change. And we're quite keen that as designers and thinkers, we actually begin to look at what might be a propositional vision to approach our built environment, or as we like to think about it, a future human habitat. So I will introduce you some of the elements that we consider to be part of the Biocene in the next few slides, and really invite you to think and maybe participate in this along with us. So it's a holistic vision. It's the idea that we want to move away from a reductionist idea of just technology that's bolted onto buildings and look at something that's more holistic. And our medium that we work with is biology. And why do we do that? We do that because biology has amazing capabilities that we just can't really replicate with conventional materials. This idea of being able to work at ambient conditions, to be able to self-heal, to regenerate, but also this idea of multifunctionality is absolutely inherent to working with biology. So here you have the example of a feather. A feather is obviously used for flight, but it's also used to attract a mate. And this idea that biology seldom does only one thing is very important to how we think and how we work within BioID. So I'd like to introduce you to just some of the systems and organisms that we work with. So we may work with the microscopic, so for instance, these beautiful sort of filamentous cyanobacteria that you see in the top corner, with products made from microbial systems. So here you can see this purple and pink on the top right. That is a set of pigments produced by soil bacteria for various different purposes, right through to multicellular organisms, so working with plant cell tissue and working with cryptograms like mosses. So we try to embrace the whole entire kind of biodiversity and kingdom of life that's accessible to us as designers. And to kind of define how we do this, so we say that we work as in a regenerative practice. We are interested in how organisms and species and biotransformations become part of a building. And the reason why, of course, is we know the urgency that we face. But rather than getting into a doom loop, we want to look at what we can do to change fundamentally the systems that we work with. So we want to give biology its full agency. We don't believe in the mechanistic view of biology. We're not putting it to work. We're actually working with it in collaboration. And we also need to embrace a time-based materiality. So a lot of work, which is in the so-called, if you like, biodesign field, presents things as products, very much finished and static. We embrace this idea that biology will change, evolve, degrade, renew over time, and that has to be part of our design language as well. So we work between two different spheres. We have the sort of scientific method, which is very much what we call process design. It's informed, it's rigorous, and it's iterative in its own way. And it's coupled with this speculative practice that might be quite conceptual, quite exploratory. The tension between these two, but also the reciprocal nature of these two processes is really what characterizes how we go about doing biointegrated design. And we realize that, there isn't really a place to train and to kind of learn these methodologies. So we set up a 2-year master's program that we run at Here East. And if you ever find yourself in London, you're very, very welcome to pop in. Here East used to be the old comms building for the Olympic Games. So we're situated on the Olympic Park. And within this one building, which was just a shell when we got it, we have retrofitted it, if you like, with a lab and with a studio and with a workshop. So we're able to have in the same space the capability to grow microorganisms, but also then be able to do advanced robotics, CNC milling, all kind of different aspects of digital fabrication. So that's really important to us, that we can link these methods together. And we enjoy a really diverse student cohort from all around the world. Nobody from Hungary yet, but we never know. So the method that we teach and that we practice ourselves fuses biology, fuses fabrication, and fuses computation and digital design. Within that, we work with data-driven systems. So we may work with environmental data, information about species or context. Within this, we then have this reciprocity between the design process and process design, which leads us to a kind of further set of experimental pieces of work, whether it's designing new tools to be able to extrude new materials or doing lab work to characterize how biology might perform in certain scaffolds and systems. And all of this takes place within a social and environmental and ecological context. And I think that is the beauty of working with architects. And I say this as a biochemical engineer, that quite often work in the engineering sciences takes place in devoid of context. And I think in future, this is how we're going to have to work, where we're very much aware of the sensitivities and regionality that we're dealing with. So that's really a kind of a brief overview of how we do things. And ecology is incredibly important to us. I don't know if any of you are familiar with this. Maybe if you come from a landscape background, you might have seen this before. But this is a triangle known as Grimes triangle. And what this says is that organisms can exist at various different levels of stress. So it could be thermal stress, like today. It could be water stress. It could be pH or salinity. They can exist at different levels of disturbance, which is maybe related to our human activity or intervention in land or landscape. But nothing can live at high stress and high disturbance, and that's precisely the environment that we create within our built environment and within our cities. So a lot of the things that we do ask this question first, which is, what can we do to facilitate life? What can we do to relax some of this stress or disturbance to enable organisms to thrive? And how can our buildings create refugia, a safe place for organisms to be able to exist? So we use and we mix and match this linguistic kind of practice. So the bioreactor in my world might look like a fermentation vessel. A bioreactor in the biointegrated design world could look like a wall made out of a porous substrate, for instance. And this is just some of the kind of material palette with which we work and with which we design. And these can span, again, a range of different types of organisms, different types of life strategies, from the kind of photosynthetic organisms that you see right through to biomaterials that are blended and extracted from other sort of post-industrial waste, right through to kind of more speculative or even through to synthetic biology. So I want to just show you a couple of case studies from our work. The first actually has nothing living in it. It's all made from residues and byproducts from other industries. So this is a project we did with a sugar company. And Tate and Lyle are one of the last heavy industries left on the Thames, and they still refine sugarcane into table sugar. And part of that process involves them injecting in carbon dioxide into this sort of sugar liquor, and they precipitate out calcium carbonate, probably known to you as chalk or limestone. But this precipitated calcium carbonate removes a lot of the organics and aromatic molecules from the sugar, so you end up with this highly refined product. We were tasked with this challenge of being able to turn this into a usable material and divert it from landfill, which is where it's currently destined. And we worked with various different biomaterial formulations, calibrating this for robotic extrusion, and then designing an end effector and pump system so that we're able to deliver this material in a precise way. And one of the interesting things when you work with biological materials is obviously their heterogeneity, the complexity of working with water as a solvent is quite challenging. This particular project, we made a formulation using various different types of biopolymers, water, and then a sort of lighter solvent that would drive off a little bit quicker so that we wouldn't see cracking. And we printed it in two stages. So we printed the first few layers of these lamella that you see on the right-hand side, and then we printed the second layer and left it to cure. So this piece was extruded onto a back made out of vacuum-formed bagasse, which is also a waste product from sugarcane refining. And then we composed a series of panels with this kind of curtain fold. We wanted this very elegant drape. And it went into an exhibition called Wastage at the Design Museum. And within this exhibition, it was interesting. It was about how design is tackling waste. And a lot of the exhibitions were quite polite, let's just say. So I made a small coaster out of some nice plastic I got out from the ocean. And then we made this giant beast of a wall, which not only had a kind of formidable physical presence, but also smelled really, really, really strongly. So you couldn't miss this at the Design Museum, because before you opened the door, you knew it was there. But I think this is actually part of the point, isn't it? If we work with waste, how we actually embrace its qualities, how we acknowledge its maybe slightly more challenging aspects. Now it's cured, it doesn't smell actually too bad at all, but it's an interesting material to work with because it's ultimately highly abundant. So that's a more inert type of system. What about the living? And we have spent quite a bit of time working on something called engineered living materials. And this idea of animate materials has captured a lot of attention. And it's not necessarily always referred to something that's living. It can refer to synthetic materials that mimic some of the qualities of aliveness, if you like. So being active, being adapted, or maybe being autonomous and being able to participate somehow. We've taken a slightly different interpretation of this. So we look at materials can be transformed by the action of something growing in it. This is the cyanobacterial living material that was developed by Prantar, who was a master's student with us, now a PhD student, and now actually going on to found his own company. And this grows using a filamentous cyanobacteria, so this sort of stringy photosynthetic organism that grows like these spaghetti strands through this silica matrix. And what happens is as it grows, it photosynthesizes. So it takes in carbon dioxide. It locks it in the material as calcium carbonate. But also, as the cells grow through this aggregate, they bind the particles together and they secrete these other molecules called EPS. And so you end up with something that is relatively strong and very lightweight. And because it is grown using light, we can then use the qualities of light to change how the material looks, to change how dense the material is, and to change some of its fundamental properties, just by understanding a little bit more about the biology and what kind of wavelengths of light these organisms respond best to. And so in this way, we kind of begin to reach a very different point where we no longer seek to make the brick. I find this is also a very problematic way of going around working with living systems that you want to recreate in exact fidelity what you're trying to replace. I think what we're advocating for is that the form or the unit is actually more resembling the way it is grown and made. And that, I think, will characterize the first set of these kind of materials that will be implemented within the built environment. They just have a different way of being integrated. So in terms of the material properties, we're quite excited about this because it's very, very insulating. It's very lightweight, but it also enables light to pass through it. So we have this kind of interesting architectural quality that enables us to think about it for perhaps interior systems where you may want privacy, but you still would like to have this element of light penetration. And a lot of people ask us, is this kind of miraculous? Are you going to lock away all this carbon? The truth is not really, actually. There's a very small amount of photosynthetic organism within this material. It's very pigmented. That's why it looks so blue. But the real benefit of this material is actually in its insulation qualities, because most of the energy that's lost within a building, or certainly within the UK, is due to poor insulation. So actually we see it as a kind of low-impact way of making something that can further mitigate energy loss. And one of the kind of interesting ways in which we work is to use computation. Don't think that's going to play, but to use computation as a way of speculating and imagining what a future built environment system might look like. So we engage quite a bit with simulation and computation, not only as a kind of form generating tool, but also as a tool to enable us to really create these immersive environments, to imagine what it might be like to inhabit such a space. And this project has gone on, it's been scaled up, so we're able to make these panels that are slightly larger now, so just over 1 meter. And you can also see the qualities of, depending on how the algae was cultured and how the material was grown, it has a different sort of aesthetic quality, and also has a different kind of light transmission quality as well. We wanted to test this in a real environment, so we have a long-term partnership with the Botanic Garden in St Andrews in Scotland, and we built this little pavilion, this little folly, which we call the Bioscope. So the brief for the project was, can you make a temporary shelter which can also be used to show a little film about biodiversity in the garden? They had an artist in residence. So we took that brief and that tiny budget, and we thought, actually what we're going to do is build a structure in which we can test and host the ELMs. So they've been up for just over a year now, looking pretty good, actually. They change over time, but they don't degrade. The garden hasn't turned into a river of algae, which is amazing. But we wanted to create this porosity. So we knew that this is not an outdoor material, so we don't want to expose it to the rain non-stop, but yet we wanted to have some level of fluidity and continuum with the outside. So this pavilion is built with this sort of waffle structure, and we had 2 sets of work that were displayed in there. So we had the cyanobacterial ELMs, and then on the right-hand side you can see some thin biohybrid screens that are made with bacteria that produce melanin, and also violacin, this other purple pigment. So we were really trying to examine how these materials perform in an uncontrolled way, but also allow people to see and experience what we were making in the lab. I think that's what is also the joy of that people can encounter it and receive it in a kind of less formal way than a gallery context, which, to be honest, when you work with living materials, is not always the right place for you. So it's just a little interior shot. It's now become apparently a very, very good spot to watch birds, because you can be in the bird hide, as people call it now, and look out on the garden and experience it in a kind of way where you don't disturb nature. So finally, I just want to talk a little bit about surfaces and systems. This is something that is, I think, incredibly important to us, is how we consider the available surface area that we have created through this incredible growth of our built environment. So I can't now remember the date off the top of my head, but the built environment is set to double. So we have only actually constructed half of what we need to make. That is incredibly problematic, because we don't have the carbon budget to make what we need to build in future. But it also suggests that we're not using the surface area in a way that we could be doing. So we had this little article in this book by Christina Pardal, where we began to look at what would happen if we started to try and activate the surface area that we've got. And here are a few projects that will somehow fuse and link together eventually. On the left-hand side, you can see what Marcus calls the poikilohydric living walls. So these are bioreceptive panels They're made out of porous, cementitious materials. He's been looking at various different formulations of cork and other additives. And the idea is to have a passive green wall system. So rather than this kind of highly mechanized, highly controlled system of irrigation, and very with an intention of having this very pristine living wall, what we want to do is look at how we can make use of the fact that some materials retain water and growing small organisms like cryptograms, with the idea of enhancing the microbiome of the built environment. A lot of times we're in very sterile places, there's a lot of dysbiosis. We're beginning to understand the role and the importance of the microbiome for our own health, but also for the health of our ecosystems. So this is a way of enhancing biodiversity in unloved or unused pockets of the built environment. And here you can see a set of panels that have been installed at a private house in Edinburgh. And here you can see them before anything has grown. So on the previous slide, we had transplanted the moss, so it gives you an idea of what it could become. And here it's, if you like, the beginning stage. And that's something we've also been thinking and writing a lot about, is how we design for each of these phases in a time-based system. Now, meanwhile, so Marcus was doing this before we met and started to work together about 10 years ago. I was fiddling around with algae, doing various different things on biofuels, and I began to work with a set of designers, and we made an experimental garden for a festival in Canada. And I wanted to think about purity. I wanted to think about organizing the algae taxonomically, showcasing them, teaching people about them. So we made this huge garden of hundreds of tubes of algae, and it was very laborious, and I'll never do it again. So, but what we began to then think about together when I started working with Marcus is how we can perhaps be inspired by some of the living systems that encapsulate algae, like the sea slug on the right-hand side. It eats algae, holds them captive, and uses them to photosynthesize so it doesn't have to feed itself at certain points in the year. So we really like this. I really aesthetically really like this too. So we began to work with water-based systems, with hydrogels, which are soft, viscous materials that hold a lot of water. So here you can see we're developing a system for robotic extrusion of these materials, and how we integrate then the living into the viscous, into the sort of water-based system, and then also examining the final form of these materials, because obviously they stay soft and pliable as long as they're hydrated, and the minute they lose water, they shrink quite a lot. But this offered us a really exciting possibility to encapsulate algae and look at, instead of moving the algae around in the water, could we recirculate water over the algae? So to flip that system, I don't know if any of you have seen the BIQ house in Hamburg. We always think about, what is the BIQ house 2.0, 3.0? What would that look like? And around the same time, we were doing a project with some partners in India looking at heavy metal contamination of water. So that's kind of, if you like, my bread and butter research. I look a lot at how microorganisms can help us deal with pollution. And we realized that bringing these huge watery vats of algae out to a site like this in Kolkata were just not going to be feasible. But yeah, it was still not going to be feasible to bring out a Cougar robotic arm and start printing all of this stuff. So we had to come up with some kind of system which was led by a former PhD student and now staff member, Shanil Malik. And she proposed this tile-based system where we encapsulate the algae in the hydrogel as before, but we use these kind of vein-like channels of these leaf-like tiles to hold the hydrogel. And this is something that can then be hand-pressed, hand-molded, but the design is, if you like, digital. So it's this blend of the high-tech and the low-tech, but then validating that through the scientific work. And the demonstrator is something that is really important to us. So we often make these kind of interstitial objects that have an element of scientific research, but also have an element of design intent. And we make them as a place where we can come together and actually think through some of these problems. And I think the demonstrator for us is a really powerful tool of opening up conversations and examining what a prototype could then become. So this was exhibited at the London Design Festival as part of the Brompton Design District in 2019. Lots of people said, oh, I really like that. Can I buy that for my garden? You could, but we liked the idea, and it was really playful to think of this is doing something very serious, but it's got an aesthetic quality. So wastewater treatment and the idea of waste is normally hidden away. It's taken away from us as fast as possible. So what if we were to make it something to be proud of? And that's the power of design. I think that's what's amazing. We made a second version of it for Beazley Designs of the Year, looking at how we can apply computation and computational design to look at changing the striations of the tiles so that we can slow down the flow of water over the surface. So now I'm going to take you to how we try and merge these things together. So we're working now on a new set of projects that try to blend this idea of bioreceptivity and also bioremediation. So this is where I think even for me personally, my aesthetic, my vision of what I think a remediation system should look like has also changed. So we've been working on a piece that's called augmented polycultures, where we were looking at the porosity of ceramic structures as a way of holding algal biofilms. So instead of mixing the algae with the hydrogel, could the algae hold itself in its own little matrix? And looking at a design that comprised of two parts, one of this body where the algae would be held and a cistern where water could be recirculated. And we showed this at the Design Museum in Hamburg. This is at the very beginning, just as it was about to get started. And over time, the algae, with the water flow, were beginning to colonize the structure. And it helps us play a little bit with notions of the grotesque, of the abject, and also kind of explore how design can make what is kind of scary or creepy, or perhaps has kind of very strong notes of aversion, how we can use design to make this into something that feels under control, and to also accept, again, this time-based change of properties. So in this work, we've been trying to bring together these elements of bioreceptivity, bioremediation, and also then the biodiversity of algae that might be there. So this is where the work is going now, to relax the constraint of purity, to accept heterogeneity, and indeed to design for that and design for change. So in the last few minutes, I'll take you outside. So this is completely uncontrolled for me. This is a fabulous thing to do, but the scariest thing I'd ever done, which is to work in a kind of field site. So this is, on the left-hand side, you can see a site in Barking Riverside in the east of London. This is one of Europe's biggest brownfield developments, and it's all based on the site of a former landfill, gasworks, sort of rubbish dump. So the soil has got a legacy of all of our anthropogenic activity: heavy metals, polyaromatic hydrocarbons, and other things. In the lab, we've been interested in how we work with microorganisms that help us degrade or sequester pollutants. But the question is, how do you deploy on a site like that? And in Barking Riverside, this is a huge and very long program with parcels of land that will become meanwhile sites. So how do we activate these landscapes while they're waiting to be developed? What can we do as a meaningful intervention? So we've been working with the idea of impact printing of soil, so using robots to create form, using environmental analysis to model how, on this particular site, rainfall and wind might affect how they dry out, and then to look at integrating the kind of form-based and fabrication-based approaches. So here you can see it being made over at Here East. Ideally, what you would do is you would fabricate on site, but I don't know if anyone's ever worked with a kind of developer or a developer that's a public-private partnership before. It's incredibly complicated. So we weren't able to bring the robot to the site, so we printed it at Here East, and then we brought it over to Barking Riverside, where we installed it. So it came on these pallets. The pallets were buried, and then it simply existed on top. So this was, if you like, the early stages of design visualization of what this temporary landscape could become. The brief we set ourselves is we wanted to create topology, because topology actually enhances biodiversity, because you create different ecological niches. And we wanted to bring almost nothing and leave nothing behind. So when they are ready to develop the site, they will simply dissolve it back into the earth. And these anthromes then begin to weather and age over time. They begin to get colonized. We did plant them. We actually had an incredibly hot summer last summer. And this site is like a moonscape. So we planted a lot of native varieties and then studied how rhizobia may or may not impact how they grow. Sort of an ongoing experiment. It's complicated. I think we learned a lot by this demonstrator, so that we may repeat it slightly differently next time. But what we also began to see is that the anthromes themselves were a habitat. So we were seeing mosses, we were seeing algae, and we were seeing other cryptogamic growth happening as the weathering process happens. So the soil at this site is very clay-rich, so it makes it easy to then do this kind of impact printing technique. But yeah, we've just published the work, so if you want to read more, it's all there. As my kind of contribution, if you like, to the field of remediation, I've seen the value of design and working with designers. It adds a quality that I think that was really absent in the world of engineering and biotechnology. So together with Deepak and Anel and a few others and Marcus, we began to write what we feel is a bit of a manifesto of how people can design more effectively when they're interested in interventions, especially to do with contaminated land or contaminated water. So I want to leave you on just a final thought, which is our vision for a future human habitat. So this is the Biocene House 1.0, which we made for London Festival of Architecture for a submission. We were shortlisted. Sadly, we didn't get to build this house. It would have been fun, but hopefully we get to build maybe a future iteration, where we wanted to really design in from the get-go our ecological systems, our living elements. So rather than this kind of plug-in or bolt-on approach, which I think has characterized the first 25 years of this century, as we've been searching for more sustainable ways to do things, we have really, and we were just talking about this at dinner last night, we have not been willing to change how we live. We really want things that are almost unobtrusive and do this miraculous job of cleaning up carbon or whatever it is that we want them to do. We wanted to design a house which had all of these things embedded, but also might invite a sort of slightly different system of living. We know that with the current density of housing, we may have to look at alternative ways of sharing and cohabitation. So rather than creating a house with rooms, we were creating spaces with functions. And this follows on a lineage of, I guess, different types of, or different forms and different typologies that we want to evolve into the Biocene house. So the idea that every surface can be utilized, everything can have a function, and rainwater, for instance, taking the surface area of a roof and being able to convert this into an activated system is very important, but how this is integrated with what happens within the house is also equally important to us too. So we've just made a second iteration of this, the Biocene House 2.0, so hopefully maybe if Marcus or somebody else comes back to Edge, we'll be able to share that with you the next time. So just want to acknowledge everybody who I work with, because it's for sure, this kind of interdisciplinary work does not happen all by itself. Marcus and I have been running BioID since 2018, and we're really fortunate to have well over 100 students that have graduated now, and going off and making waves in the world, which I think is a really wonderful thing too. So thank you once again for having me. And yeah, happy to take any questions now. Maybe no time, but we can certainly catch up over coffee or later today. Thank you.